inviting me today to present our work on the genetic diversity of Campylobacter larvae in a One Health context, with a focus on shellfish, wild birds, surface waters, and the associated human health risk. So as you know, on the watershed there are several sources of fecal pollution that comes from, for example, from bovine breeding, manure breeding, wastewater treatment plants related to the human activities, also direct discharge from the houses, where you can also have the porcine breedings related to animal culture, and all these sources can go through rivers. Likely during wet weather, during rain, so you have a runoff of the surfaces, and all this fecal material can go through the rivers and to the sea. Once arrived at the sea, so we have central risks for the users of course, for persons who have recreative activities like bathing and fishing. But for today's talk, we will focus on the shellfish harvesting areas and the shellfish that are after consumed by the population. So when these shellfish are contaminated, there is a central risk for consumers, and we can have what we know of collective foodborne poisoning. So two major effects we can have. The first one is the economic consequences, closer declinization of the shellfish harvesting areas or the bathing areas, and as I said, the human health risks. So the objectives of our work were to first have an idea of the identity of the sources. How can we identify each source of pollution in the upstream catchment? And after this, we focused on a bacterial species, which is Campylobacter, and try to see its presence and in which emergence in each compartment of the catchment. So this is for the group, the genus of Campylobacter, and surprisingly we found that there is a species or a group of species which was dominant, which is the group of Campylobacter larvae. So we focused our work on this group of species to study the genetic diversity, in shellfish and also in wild birds. So to do this, the first step was to identify the sources of fecal pollution, and to do this, we used the 16S metataxonomic method. So I said before, we know that in every source of pollution, we have what we call the fecal undicolor bacteria, which are E. coli and Enterococcus. So they are common to every fecal pollution. But the problem is when we monitor this microorganism, we can't identify exactly what is the exact source of pollution. So in the recent years, there was development of markers. Here we have a non-exhaustive list of markers for human ruminant, cattle, dog, and avian markers. And simply it represents a bacterial species which is specific to each pollution, each source. And so researchers developed qPCR primers to amplify specifically those targets. And for example, if we use the HF183 and we get positive results, we are pretty sure that the source of pollution comes from wastewaters, for example. And so as an example, so as I said, in each solution you will have E. coli, which is present in each solution, but we have markers colored here in yellow, red, and blue. And so when we have a pollution at a specific time, here, for example, as I said, we can really be sure that the pollution comes from the human. So up to date, the markers developed are mainly for ruminants, cattle, but there is already no marker for the wild birds. So this work was also meant to try to develop such markers. So to do this, we've done samplings in different regions of the Atlantic side of France. And so we got about 565 samples from almost 12 wild bird species and also Lake's chicken also for the domestic birds. So for the metabarcoding technique, the strategy we used is to target the RNA ribose mechanoresorry 16S. And we used the short read technology to do this, so we were not able to amplify the whole gen. So we targeted the V3, V4 region using specific primers. And so using this strategy, we can identify at the right of the slide. We can say using this we can have barcodes for each species or genus, and thus identify the bacterial communities in our samples. There are other techniques that are already used today. So, for example, targeting the complete marker. So we can, with the long read techniques, we can amplify the whole 16S gene, and with this we can easily identify the species. Or another technique, but it's more expensive, is to use the shotgun sequencing, where we will sequence all the genomes of each sample. So here, an example of a result that we got for the data for the whole data set of the wild birds samples. And what we can say is that we have four dominant phylums: Primicus, Proteobacteria, Actinobacteria, and Fusobacteria. But also we detected Bacteroidetes, which I forgot to say it. So Bacteroidetes is a marker for the human and cattle social pollution, and we detected some Bacteroidetes in our samples of wild birds. So this confirms that the markers are not 100% efficient in identifying each source. So in no detail, I did not give a lot of details, but here is a map of the results we got. So the diversity at the genus level in our samples, they are by bird species. And so what we can see here, that there is a large diversity of the genera that we found. And thank you. And another observation we got, for example, here I highlight the Cattylococcus genera, which we found in several wild bird species. And initially, researchers who developed the GFD marker, it was meant to identify only the pollution from seagull. But the problem is, using this high-throughput sequencing strategy, we confirmed that this Cattylococcus marker can be found in other wild bird species, and so this can make mismatch between the source identification between each wild bird. And from this data, we see also we got also, so we detected pathogen signatures and mainly with Campylobacter and Helicobacter signatures. In parallel, we've done the same. We've done the same strategy. So the metataxonomic profiling of samples from the other sources, so the domestic birds, cattle, and the wastewater treatment plant. And so the idea after this is to remember, it's to identify specific markers for wild birds. So when we compare all this data set together, we can clearly see that there is microbial signatures which are different between each source, and we see that and using this data set, so I don't detail the bioinformatic approach, but using this, by comparing all the data set, we identified the group of genus or bacterial genus which was specifically present only in UTS1. So here we've done initial tests using qRT-PCR, and it's pretty promising marker with up to 75 percent sensitivity and up to 90% specificity, which is pretty good. But I think we need to do more experiment to get higher percentages of sensitivity and specificity. Yes, so this is the first way to use this data. The other thing that we can do with this metataxonomic data is we can, so at the left you have the signatures of each source. For example, I put here a human, cattle, birds. On the other hand, for example, we will take the water from the seawater. We will do also the metataxonomic data and use bioinformatics using a tool called SUSTracker. By combining both data sets, we can predict, it's not perfect, but we can predict that we have certain percentages of pollution coming from each source in our sample. Yes. Always using this data set, we specifically searched for the presence of, so we selected a list of pathogens, and we tried to look for the most abundant genus pathogen in our data set. And for wild birds, we found that it was the second most abundant pathogenic genus in our data set. So for the next parts of the presentation, I will focus on this genus. So to study this bacteria, we used at this time the whole sequencing, genome sequencing approach. A quick presentation of the Campylobacter genus. So it belongs to the Ipsilobacteria. It's a gram-negative bacteria, non-spore forming, motile. It is the most common causative agent of gastroenteritis in humans. It's predominant in many sources, from wild birds, domestic, livestock animals, etc. And there is about 33 species within the genus, and the most studied species are three, Jejuni, coli, and Larry. So for the Larry group, it's a member of the thermotolerant group of Campylobacter. It's more difficult to culture than coli and Jejuni. It was detected in sporadic cases, less than Jejuni coli, but sporadic cases exist for this group of species. In the opposite of coli Jejuni, C. Larrae can more easily spread and grow in saline water, so and shorebirds also. He was also more halotolerant, so to the salt waters. For the genotyping of this species, as you can see, I will detail this in the next slides. So one genotyping tool is the sequence typing. And for example, for the whole group of C. Larrae, we have up to date only 342 sequence types versus up to 15,000 STs for Jejuni and coli. So to achieve our work, we've done a series of samplings at the Baie-de-la-Fresnaye catchment, which is connected with 4 rivers, and there's about 9,000 inhabitants in this region, and this region is also known for an intensive livestock farming. So we may have sources of pollution from these farmings and also from the inhabitants of the region. So we've done a series of samplings for shellfish, oysters, mussels, and also cockles, surface waters, of course, wild birds in the region, and the human isolates from the wastewater treatment plant. So for the results that we've got, in average, we observed that about 39% of the shellfish batches were positive to Campylobacter. And you have here the other results. For example, for river waters, for the four rivers, we have a high prevalence with up to 85% of positive samples. And for the shellfish, it's about from 37 to 45% positive samples in our samples. Something also that we observed for the culture method, we didn't find a clear correlation between the presence of Campylobacter and E. coli, which means that the E. coli is the indicator of pollution, is not always effective if we can check the presence of pathogens. Another observation that we got, so we've done all the samplings during the year, and what we observed is surprisingly there was a significant seasonal effect where we got a lot of Campylobacters during cold weather, so the winter and autumn versus spring and summer. Then also, if we have a look of the prevalence of the, so if now we look for the prevalence of each species in our samples, surprisingly, we found that Jejuni and coli were more present in the rivers, so in the less saline water. But Larry, as I said before, he likes more saline water, so it was more prevalent in seawater and in our shellfish samples. For wild birds, we have also a differential. So first, we had the dominance of Ciliary in all samples, and then we have the Jejuni and coli and another species which was less predominant, which was Piroides. And so here you have an example of some groups of wild birds with the distribution of each bacterial species. So at this time we have a nice view of the prevalence of the species in the different sources. The next step is to identify the genotypes. So as you know, we can, on the Petri dish, we can have the same phenotype, the same colonies, but maybe there are mostly what we have are two different strains with different genomes. So as I said before, we can use the 16S to genotype our isolates, but using this marker, we will have we can identify only the species. Another tool that we can use for genotyping is the multi-locus sequence typing. The approach is to sequence at the same time 6 to 8 genes. And so when doing this for each strain, each gene will have what we call an allele. So we will give to each gene an allele, and the combination of the codes for each allele give us the ST or sequence type. And at this time also, you can have two strains having the same MLST, so it's, at this time also we can be sure that it's a clone or different strains. So to do this, we use the whole genome sequencing. And so at this time, so we will really sequence the whole genome, identifying the chromosome, the involuntary plasmids, and at this time we can clearly separate or identify the different strains of our collection. So to do this, you have today mainly two strategies, the short reads and the long reads. The gold standard for short reads is Illumina, which gives what we call a draft genome. So the genome is, we have gaps in the genome, so it's not a complete genome. So we don't have a clear view of the genome. Here, for example, we have the great chromosome and two small, which should be plasmids. For the second technique, so the long reads, using the MinION from Oxford Nanopore Technologies. So here we will have a better view, as it's the same genome. So here we have the chromosome, but it's linear, and we have also these two small, which should be plasmids. So we have a better view, but it's not really yet clear. And using bioinformatic tools like Unicycler, we can combine both data sets. And using this, we will have a clean chromosome, circular chromosome, and the plasmids with the complete view of our genome. So using this strategy, we sequenced the whole data set, the whole strains that we isolated from the watershed. And as you can see, for C. jejuni and C. coli, we've done some phylogenetic analysis, and you have only two closed clusters. But when we look for the Ciliary group, we have the highest, higher diversity with the identification of the Ciliary group, but also what should be maybe also a new species that I will talk about in the next slide. As I talked about the MLST, so use this to genotype our strains, and it gives the same clusters, clades, as for the whole genome phylogeny. So here for the ciliary group, we identified 40 different sequence types, and this led us also to identify the same clones that are present in different sources, seawater, shellfish, and human, and this can explain that some clones can already come from shellfish and contain humans, so that's why we detect the same clones in the different sources. Here is the pangenomic analysis of the whole dataset. So as you can see at the left, so you have all the samples, and here it's a heat map for the similarity between genomes. So simply it's a blast at the genome level, and using this we can grab the genomes by groups and it identifies so to each strain belong to each cluster. So here you have Coli indigene at up and the ciliary group and at the bottom. And on the right slide you have the number of genes for our samples, and what you can see is that Coli indigene share a high number of genes, up to 80%, 28%, sorry, while they share only 60 or 70% respectively with ciliary. And so using this analysis, we found that there was a clade which was phylogically distant from the ciliary group, so we were interested in this clade. So when we analyzed this clade, there were nine isolates. Sorry, 2 were isolated from every water, 6 from every water, and 3 from human stools. And so doing some phylogenetic analysis, we identified that we concluded that this 9 strain belongs to a new species that we call the Campylobacter americus. And so this extends the phylogeny of the group of Campylobacter. Just a quick reminder that we used also the hybrid strategy of sequences using Illumina and Oxford Nanopore for one strain of this new species. And so using this strategy, we've done different analyses, and we selected, for example, the second result, identifying clearly the chromosome and both plasmids. What we can do also with the genomic data is we can screen the genes of interest. And since we are working with the pathogenic bacteria, so we can screen the genome, for variant genes, for example, genes coding for flagella, pili, adhesion, biofilm, secretion systems, toxins. On the other hand, we were also interested in genes coding for antibiotic resistance and the efflux pumps. And all these genes can also be mobile. So this is the interest of the hybrid strategy of sequencing. So we can easily detect the plasmids and also screen specifically the plasmids for these genes. So using this, I will not go in details, but just to show you that we screened the 3 species, and what we can see is that Coli indigene are known to be pathogenic. It's established, but Leroy, this is initial data in literature. There are only some studies, and we clearly see that also the ciliary group contain genes for antibiotic resistance, which make it also a possible an emergent pathogen. And this, for example, for the antibiotic resistant genes, they are known to circulate in the population also by the horizontal gene transfer between species of different genera. Here is the same result, but for the variant strains, and also we can see that the ciliary group also can harbor several genes that were previously confirmed as having a toxic effect on in vitro for Coli and antigenic. Yes. So for the conclusion, and if at the end, so I think we don't eat shellfish anymore because of all this pollution, but of course, there are controls of the shellfish to guarantee the food security of the consumers. So the real take-home message is that from our results and the bibliography data, clearly Campylobacter is the dominant bacterial genus in catchment. Ciliary is clearly dominant in shellfish and seabirds, and more research work is needed to get a higher number of sequence types, to identify maybe other species, and to get more insights in the pathogenicity of this Campylobacter species. Of course, you have to keep in mind that we use limited detection methods for the culture methods. So maybe sometimes we have false positive or false negatives because the ciliary group is very diverse metabolically. So maybe we at the lab we use standard Campylobacter specific culture media. But maybe we can't culture the new species that we have we have to discover in the future. And to finish, so to make connection between the two parts, the idea at the end is to try to connect the detection of sources using the microbacillus tracking, and to be sufficiently efficient to really predict the presence also at the same time of Campylobacter pathogens in our samples. So at the end, I want to thank all the partners of this great project and you for your attention. Thank you.